Is funded by a variety of federal, foundation, and institutional sources, and I have no financial disclosures or conflicts of interest to report. There are three objectives for this brief talk. First, we'll discuss how the Connectome scanner has allowed us to advance knowledge about recovery from severe traumatic brain injury. Second, how the scanner is also helping to generate insights into how repeated blast exposure affects the brain health of military service members and veterans. And then finally, a theme that will run throughout this talk is how a network-based autopsy of the human brain that leverages ex vivo MRI using the Connectome scanner is making fundamental contributions to brain health and pathophysiology. First, traumatic coma. This young man, Doug, was the first patient with traumatic brain injury I took care of as a medical student back in 2006. Also the first patient to be scanned on the Connectome 1 scanner back in the day when it was implemented in the early 2010s. And Doug's story, just to give you a sense of the motivation for this work, is a college junior, cyclist for the Drexel cycling team. This is all shared with his consent. And he gets hit by a car in West Philadelphia, hit and run, comatose for 14 days, completely unresponsive. The medical team told his parents that he would likely die from his injuries in the ICU, and if he survived, he would probably end up in a vegetative state. This is a picture of Doug taken by his parents in the ICU, and that prognosis that I just shared was based upon this low-resolution gradient echo scan, where each of these hypo-intense lesions represented hemorrhages within his brain. The shearing forces at the moment of impact tore the blood vessels and presumably tore nearby axons, disconnecting his neural networks and causing a coma. And what was of particular prognostic relevance was this single lesion in his brainstem, the brainstem, as you know, being the battery that sends ascending activating signals to stimulate the rest of the brain. These were the reasons for the poor prognosis. To cut to the punchline, after a lengthy inpatient and outpatient rehabilitation, he gets back on his bike, rides coast to coast from Cali back to West Philadelphia, stopping in rehabilitation centers in every state to raise awareness for TBI recovery. There's a picture of him taken several years later at the exact spot where he was hit in West Philly, wearing his Drexel uniform. I still get the chills and find this incredibly inspiring every time I look at it. So how did we get it so wrong? We, the clinicians, told his parents that he would have a terrible outcome. Many patients like this, their families will withdraw life-sustaining therapy in the ICU. That is the #1 cause of death in this patient population. 70% of all patients who die in the ICU after a severe traumatic brain injury, it is because of the withdrawal of life-sustaining therapy. Thankfully, Doug's parents didn't listen to us. And the question that we asked when we brought him to the Connectome scanner in 2012 is, were there brainstem connections that we couldn't see with that gradient echo, but that could have been detected and that could inform the development of more accurate prognostic models? Just to reorient us, here is that gradient echo scan from day 7 in the ICU. We re-imaged him with susceptibility imaging on Connectome 1. There's that same lesion because of the hemosiderin deposition. It happens to overlap with a key arousal nucleus called the peduncular tegmental nucleus, shown here in turquoise. And what we're looking at in these next few panels, here from a posterior perspective, is how that hemorrhage modeled in red overlaps with this key nucleus. And then in the bottom left and bottom right panels, we're looking from an inferior and posterior view, respectively, at the diffusion tractography data, the tracks within his brainstem that we detected many years later after his recovery. And the key observations are as follows. First, yes, there does appear to be some asymmetry. The hemorrhage may have caused some local disruption of tracks, but there's still many pathways that are intact, pathways that may have been the basis for his long-term recovery. I want to give a shout out here to Jen McNabb, who was a key collaborator in this project and performed this work with me and our team early on in the 2010s. Our goal was to translate these new findings that we developed on the Connectome 1 scanner into the ICU scanner. We have a 3T Skyra, doesn't have the same magnetic gradients, but based on the proof of principle in Connectome 1, we implemented ultra-high resolution diffusion imaging into the ICU on Lunder 6 at the main campus, and here are some representative results. We're looking from a lateral perspective, first at a control, then at a different patient with severe traumatic brain injury, where we performed this imaging while he was still in the ICU, and it's the same story. Yes, there are some disconnections. There is a difference between the patient and the control. but we again see preserved tracks connecting with the cortex that may provide the basis for ongoing recovery. And indeed, this young man who was hit by a car recovers consciousness, communication, gets married, rejoins the workforce as a physical therapist. We're beginning to see the pathways that allow patients to recover consciousness based on these key insights initially generated in Connectome 1. I want to give credit here to Dr. Sam Snyder, who played a key role in these brain mapping studies in the ICU. Taking a step back, as I prepared this talk, it was a trip down memory lane. As I mentioned, Jen McNabb and I performed a lot of this work together. And I just want to highlight the team science that emerged from this incredible resource that we all had access to, Connectome 1 back in the day, Connectome 2 today. This is Doug when he traveled to the scanner here sitting at the Connectome Bay in Bay 8. Here's Jen and I scanning some ex vivo brains circa 2010 or 11. All of you know Andre Vanderkow and the incredibly important contributions he has made to every step of this work. Lila Zale, Hannah Kinney, our pathology collaborator, and then, of course, Thomas Witzel and John Palomini, for those who remember, our old friends. Just wanted to acknowledge how fulfilling it has been to do this work, not just on behalf of our patients, but also in collaboration with such a great team. For the second half of this brief talk, I'm going to transition to a different topic, where our goal has been to detect or to see the invisible wounds of war in military service members and veterans. By way of background, we have been focused on the Special Operations Forces community, such as Navy SEALs and Army Rangers, who are frequently exposed to explosive blasts, mostly during training and also during combat. These repeated blast exposures can be associated with a broad spectrum of cognitive, psychological, and physical symptoms, but there is no way currently, with today's clinical conventional MRI, to detect the brain injury that is believed to contribute to these symptoms. For the individual operators who are protecting and defending our country, this means that they are often told, and we have heard this story time and time again, hey man, it's all in your head, get back out there, we don't see anything on the scan. Or maybe it's depression or some psychiatric illness that you came to the service with. People go back into training or combat when their brains are vulnerable. That exacerbates their injuries. And the misdiagnosis or lack of diagnosis can cause despair, shame, moral injury, and in the most tragic circumstances, these individuals have died by suicide, only to then be found at autopsy to have brains full of scarring, believed to be from these blast forces that were completely missed by diagnostic tests. At the level of our force, when you think about national security, the lack of a diagnostic test can undermine the readiness and performance of our most elite troops. It's a barrier to recruitment and can also compromise retention. So let's be clear about what we are looking for here. Many of us, when we think of blast exposure in the military, we think of a roadside bomb in Afghanistan or Iraq, big IED blast goes off, and somebody has symptoms at that moment. A traumatic brain injury from a blast exposure. That is not what we are trying to detect here. Rather, we are attempting to detect the effects of repeated subconcussive blasts, like those shown here in the video, mostly incurred during training, where these individuals over and over and over again every day are exposed to thousands upon thousands of blasts from breaching heavy artillery and other types of weapons. We were fortunate to partner with the United States Special Operations Command as part of this effort. I see Kiara Mathe here, who played a key leadership role in this study from its inception in 2019. These are some of the leaders from Special Operations Command who came to visit us. General Clark, after his visit, testified to Congress that the MGH Martino Center, and he actually gave a specific shout out to ex vivo MRI, which was particularly cool. He talked about how we were a key hub in their research efforts. And then more recently, General Fenton on 60 Minutes talked about this research effort as a sacred obligation to the force. We have been scanning active duty Navy SEALs. We are attempting to enroll 100 over the next few years. The preliminary results from that study are currently under control by SOCOM. I apologize that I'm not able to present the in vivo results to you. And again, that is something that is out of my control. But what we're hoping to achieve by the end of this study, and I look forward to being able to present the collective results within the next few years, is a risk matrix that we can give to commanders and team leaders to give them a sense of the readiness and the operational performance of their teams. And the way we'll do this is by combining imaging biomarkers, blood biomarkers, and symptoms, such that individuals who have abnormal biomarkers, I'll show you an example of what that would look like in a moment, and those who are symptomatic would be considered high risk as having possible repeated blast brain injury. Why possible? Why not definite? This remains a diagnosis that can only be made at autopsy, similar to chronic traumatic encephalopathy with contact sport athletes like NFL players. And so what we are attempting to do now is to link microscopic findings at autopsy to ex vivo MRI to in vivo MRI, so that what is currently considered a possible diagnosis will ultimately be confirmed. We're doing this with an ultra high resolution ex vivo MRI protocol at both 7T and using Connectome 2.0. We then section and stain every specimen with our pathological analyses targeted to the well-established nodes of canonical neural networks, as shown here, part of a large multi-center collaboration with the team shown on the right side of the slide. And the conceptual basis for this approach is that the ex vivo MRI serves as a methodologic bridge that links gold standard observations made with pathology to observations made pre-mortem with MRI, EEG, or cognitive evaluations. We have been fortunate to partner with a team that is creating spatial alignment techniques for the gold standard pathology data to the MRI data. Here we are zooming in on a human hemisphere scanned ex vivo at higher and higher resolutions from 250 microns to 50 to 5 microns using optical coherence tomography to submicron Nissl stained tissue, where every image that we're looking at in this video is precisely aligned to the one that came before it, allowing us to truly go from microscope to ex vivo MRI, ultimately to in vivo MRI, to inferences made at the bedside. And I want to thank the team shown here who have played a critical role in developing these types of alignment techniques. Our key preliminary data from the post-mortem scans on Connectome 2.0. Here we're looking from a lateral perspective, first at 2 civilian brains, from individuals who had severe TBI from car accidents and died several years later. We have peeled off the cortex and we are focused on the gray-white matter junction, the anatomic region that is particularly susceptible to scarring from repeated blast exposure based on the pathology data. Now here are 2 Navy SEAL veterans, scanned post-mortem, where you can see on this heat map at the gray-white junction, lower F-soma. This speaks to the Sandy imaging that was discussed by several of the prior presenters. I want to acknowledge Dong-Suk Sung and Susie Huang, who have been leading these analyses. Now, for full transparency, these are preliminary results. This is a small sample size, and we are waiting, perhaps this week, for the correlative pathology data to understand why there are these hotspots of lower F-soma at the gray-white junction in SEAL veterans as compared to civilians. So any interpretation that I give here is purely speculative. It's possible that the scarring has led to decreased numbers of neurons and therefore decreased isotropic diffusion within soma. Again, that is a preliminary interpretation, but the idea is that we're beginning to move closer to seeing the invisible wounds of war with this technology. To conclude, we do this work in honor of the many brave men and women who serve our country. These are 3 individuals, their stories have all been made public, who after blast exposure developed symptoms, sought care, had normal conventional MRI scans, died by suicide, and brains were found to have extensive pathology at autopsy. Every one of their family members, their Gold Star families, have said that they believe their loved ones would be alive today if their wounds had been seen during life and if they had the validation of knowing that there was something going on in their brain that caused their symptoms. So we hope to honor their memory and their families with this work. Appreciate all the collaborators in this room who have contributed to it, and happy to take any questions if we have time.
Thank you so much, Brian. Yeah, for that amazing and inspiring talk. Are there any questions in the audience? Yeah, Shaheen.
The previous slide, you showed that, the pattern of, yes, exactly. So the pattern of, brain changes in civilians versus SFPR looks to be opposite. So how do you know? Because it doesn't mean that, those soldiers are showing a more exaggerated effect. It's exactly opposite to them. How do you interpret that?
Great question. Short answer is we don't know. This is still preliminary. We have 3 control brains who died of non-neurologic causes waiting to be scanned on Connectome 2. And I think that's going to be a key part of the story because clearly comparing 2 injured populations to one another makes the interpretation difficult. But they're due based on pathology data. The scarring that occurs at the gray-white junction, as indicated by GFAP stains in military personnel, is very different from the axonal pathology that's more widespread and diffuse in civilians with rotational forces, like during a car accident. So my very preliminary interpretation is that there's something there, the mechanistic differences likely explain what we're seeing here with these different effects, but what is happening at the tissue level until we have the path data and until we have controls, I hesitate to speculate too much. Hope to have a better answer to that question in a couple months.